set up house in your lungs and form tubercles. You would be in a latent stage of tuberculosis. When the tubercles burst open, the bacteria escape. That's when you're in active stage of tuberculosis. The bacteria can enter your blood system and travel to the rest of your body and infect other parts. And your droplets are going to be more infectious to other people at that time. So, make sure you read about tuberculosis. When you go in the nursing program, they're not going to stop and educate you about tuberculosis. They're going to think you've already had that. I can't tell you. I've had a number of emails from students, oh my God, I don't remember the thing about tuberculosis. Yeah, that's why I'm teaching you about it now. Because you're going to get in the nursing program and you're going to have to understand what tuberculosis is and why it's so dangerous. And I'll show you a movie on that probably tomorrow. Okay. Here's a picture of an x-ray. In your book, somebody has tubercles from tuberculosis in the lungs. Many times the tubercles will actually be up higher in the lungs because when you inhale and exhale, the lower part of your lungs does more dispelling of the oxygen. This upper part tends to kind of stay filled with a little bit of oxygen all the time. So there's actually more oxygen in your lungs up here. So for some people, the tubercles will be up higher in the lungs. But either way, tuberculosis is a lower respiratory tract infection. It's not in your nares and throat, it's in your lungs. So if I want to test you to see if you're actively infectious, if you have tuberculosis and could possibly be infecting other people, I'm going to test that by examining your sputum. Not your spittle, that's spittle. I want sputum, I want that, that gross stuff. And so respiratory therapists will come around and take a sputum sample from the patient, take it to the lab, and then they'll look at the sputum sample and do an acid-fast stain on it. If they find acid-fast bacilli in the sputum, that would indicate that you've got tuberculosis or leprosy bacteria, because leprosy is droplet infection also, in your lungs. And by the way, your textbook chapter also goes into leprosy. Here's a beautiful picture of the leper's hand. Sad, isn't it? And so what we're going to do today is imitate one of the tests they might do on a patient to see if he's actively infectious. We're going to use an AFB check slide. So sometimes they'll call these bacteria, Mycobacterium tuberculosis, sometimes they call them the tubercle bacilli because they're bacillus in shape and they form tubercles. Sometimes they'll call them acid-fast bacilli, and that's what AFB stands for. Acid-fast bacillus check. We're checking for acid-fast bacilli. If you read the pamphlet that comes with the package, or if you go on the internet, it'll say, definition, AFB check is a specially designed microscope slide intended to be part of a quality control program to monitor stains and staining techniques. That's not what we're going to use it for. We're going to use it to check for acid-fast bacilli in sputum. Okay, we're actually not going to use our sputum because that would be disgusting. We're going to just do a mouth scraping, okay? And then it tells you on each slide, it contains one section, it says oval, one round section of the positive control, heat-fixed acid-fast positive control Mycobacterium kansasi. It's too dangerous to put Mycobacterium tuberculosis here, so they put a less pathogenic, but still pathogenic, bacterium here. Your positive control is Mycobacterium kansasi. That's why everything is red. These slides contain a pathogen, Mycobacterium kansasi. Your negative control is E. coli. Those two cultures are already on your slide. You're going to stain them. And you would stain your patient's sputum sample alongside. So it's like a little experiment. Here's your patient's sputum. You're going to compare what you see in your patient's sputum to what you see here. If you've got some that looks like this, that might indicate your patient's got acid-fast bacilli in his sputum, mycobacterium in his sputum, because they match these mycobacteria, right? E. coli, you don't ever have E. coli in your mouth unless you've been doing something kinky. Or kissing your cats like I do. I'm like, oh, that's lovely. They go in the litter box, their feet are in the litter box, they poop in the litter box, they lick their butt, they lick their whole body, and then I'm like, yeah, come sleep with mommy. So, yeah, but yet I get nervous when I go out to dinner. Okay, whatever. I think I'm immune to every cat germ. Okay, so your E. coli, though, is a control. If the E. coli don't come out the way they're supposed to, and if this doesn't come out the way it's supposed to, then this is not reliable. It's a do-over. See what I mean? So you have a positive and negative control to show you if your staining procedure is correct. If you didn't do your stain properly, these are not going to come out the right way. And if those don't come out the right way, how can you consider what this result is? It's invalid. Throw it out, do it over. Got it? Okay. When you do your smear, you're going to stain, after you put your smear in here, you're going to stain all 3 areas of the slide. You're going to treat them as equally as you possibly can. Okay? So when you put a drop of stain over here, oh, a few drops over here. Then you're going to rinse. You're going to decolorize here, here, decolorize here. Rinse. Then you're going to put a drop of stain, a couple of drops of stain. And then you're going to rinse. So you've got to treat all 3 areas as much the same as you can. Just accounting for the fact that the little circles are smaller than your big oval. Okay? Now the acid-fast stain procedure is a differential stain. Oh, you know what? I have a little video clip of this. It's going to differentiate between acid-fast bacteria, bacteria that have an acid-fast cell wall, that means it has waxes in it, mycolic acid waxes, from bacteria that do not have the waxes in the wall. They're non-acid-fast, or they're acid-fast negative. The only ones that are acid-fast are going to be genus Mycobacterium, maybe genus Nocardia. Okay. Well, let me show you how to make your slide, and then I'll play with the movie, and if it looks good, we'll show the movie. Here's a movie about differential stains, a little clip. Okay, so what you're going to do is take an acid-fast bacillus slide, an AFB check slide, and it's in an envelope. And it's red because it contains a pathogen. So do not lick your fingers. Do not lick the slide. Handle it carefully. So open your envelope from the top and then make it like a little pooch. Pooch it out and then reach in with your clothespin and you'll grab the top of your slide. See? And now my slide looks kind of like my drawing here. It's got a little area for positive control, a little area for negative control. Actually, the little plus sign is up here. So that's your positive control and that's your negative control. And then it's got an empty oval. Okay? Save your little envelope. When you're done, you can put your slide in the envelope, throw it in the biohazard box. Or if you put this in the sharps container, throw your envelope in the biohazard box because it's been touching the pathogenic area. So this is contaminated. So that goes in the biohazard box. Now you're going to need a swab. So swabs, there's a right way and a wrong way to open swabs. You got to find the leaves here, and open it from the handle end. Oh, there, that didn't work out nicely. You're going to peel it apart and the handle will stick out. Because you want to grab it by the handle, not by the swab part. This part is sterile. It's wrapped because it's sterile. So just take a scraping. In my early morning class, I got like 3 squamous cells. I'm like, really? Like, scrape the freaking mouth, okay? So I usually call it a tooth scraping because you should scrape up along your gum line where you have like dental plaque and stuff. Watch me knock a cap off. But also, you can rub it inside your cheek or over your tongue if you want to. Oh, tongue, hey, go back. I had a chocolate shake just a few minutes ago. All right. All you have to do is roll it onto your slide. So press it and roll it on. And you can roll a row, then roll the next, and So you can roll it on 3 times, fill up that space. I want you to have enough cells to look at. Some people did, I don't know what they did, but they got like 3 cells on there. Okay, this is technically biohazardous waste because we're at school. So put it back in your little envelope and it goes in the biohazard box. Now this thing is going to dry in 2 seconds because it's stuck with spit and goop, basically. And then you're going to have to heat fix it. So I'm actually going to heat fix mine, so look out, honey, before I catch